Hello, everybody. It's Miss Peachy again, and I am here for your Biology 8, Unit 2, Lesson 10 on genetic research. This one I think isn't such a difficult lesson. I feel like we're kind of taking a step back from the molecular biology, which is good because that stuff can be kind of hard. But we are going to be looking at genes and kind of looking at how we can predict kind of what the offspring are going to have, what kind of characteristics and what kind of traits, using kind of some of the rules of inheritance. So again, I point you to my website where I will have a copy of this notes guide for you for this particular lesson. So hopefully that'll help you kind of wheedle out the key concepts and things like that. So again, over on this side of the screen is the actual lesson viewer, and kind of down below me and to my other side, so backwards for me here, is going to be kind of the information and the notes that we'll be going through today. So a lot of vocabulary here, but I think it's a bit easier and maybe a bit more familiar vocabulary to you. Essentially talking about how, even though back in history, people didn't really know a lot about transcription and translation and protein synthesis, that process, which we call, I haven't used this term before, but it's oftentimes called the central dogma of biology because it's such an important part of biology. Even though we didn't know about this, they didn't know about this in the past, they did recognize that traits and characteristics were inheritable, that you could inherit traits from parent to offspring, in people, in animals, in plants, that you could kind of breed different organisms to be able to elicit certain traits. So some of that knowledge was there, even though the understanding of how it worked wasn't there. And so talk a little bit about a monk by the name of Gregor Mendel, who worked a lot with plants in the 19th century. He spent a lot of time crossbreeding plants and kind of figuring out some of the rules of inheritance. And he did a lot of work with pea plants. He would pollinate them. He would try to get specific characteristics and be able to kind of determine like which characteristics were kind of more often seen and which ones weren't. And he established some of these different rules, which we'll talk about. He did recognize that there were different, I called them different variations of specific traits. He didn't know what a gene was, but we would say it's a different variation of a gene, and that sometimes you could have one variation and sometimes you could have a different variation. And he called these variations, are called alleles. I'm not sure if Gregor Mendel called them alleles or if they were called alleles later. For example, one of the alleles for the gene that causes coloration of flowers in pea plants could code for maybe purple flowers or white flowers, so different coloration in the flowers, right? So that's an allele. He also discovered that some traits were considered to be dominant and others were recessive. And by dominant, meaning that if two organisms were bred together and one of them had the dominant trait, that would be the one you would see in the next generation. It would always mask or cover up the recessive trait. So he established rules of dominance and recessiveness. Dominant meaning that allele is something that will, if two different alleles are present, a dominant and a recessive one, the dominant one will always show and the recessive one will not. A couple other terms we should be familiar with here. Mendel had different terminology for different things, but what we know is that there are always two alleles for every gene. Okay, so remember that DNA is double-stranded. So you've got two alleles for every gene. One allele you inherit from your mother and one allele you inherit from your father. Because there's two alleles for every gene, sometimes you have those two alleles are exactly the same. So if the two alleles are the same, then we call that homozygous. So let's put that on the board. And I decided That typing is the way to go in my last video, even though even that one is not my favorite. So we'll call this homozygous. That should be a 2. 2 alleles for a gene are the same. Now, by the way, Mendel often referred to, this does not say this in the lesson, but he often refers to homozygous as being purebred or true breeding. That's kind of a word that he would have used. So I'm just going to put, and that's important because, we don't like to talk about purebred with people, obviously. It can kind of have some negative connotations, but when we're talking about animals and stuff like that, we oftentimes refer to purebred animals, right? That's something that people oftentimes use in the animal industry when you're making like dogs and breeding horses and things. I don't know a lot about that stuff, but they definitely use the term purebred. And then we have heterozygous. And this is when the 2 alleles are different. And remember, you always have 2 because you've got your one from you inherit from your mother and one you inherit from your father. Mendel referred to these as hybrids. These are hybrids, okay? So what he did was he experimented with breeding pea plants. He would fertilize them over and over and over again until he would get some that would always produce the same traits, and he would call those true breeding. So every time he would pollinate them, he would self-pollinate them. They would continuously produce the same traits, so then he would conclude that those were true breeding or homozygous. He had no way of testing the genetics for the plant, but the fact that generation after generation got the exact same coloration or whatever that characteristic happened to be, he would then conclude them to be purebred or true breeding. All right, so what he did was he essentially, like I said, created kind of groups of plants that he was able to say, this is a pure breeding for purple flowers. This one's pure breeding for white flowers. This one's pure breeding for wrinkled peas. This one's pure breeding for smooth peas. I mean, he had a lot of time on his hands, so he was able to do a lot of these different experimentations over many, many years. You also have a few more terms that we're going to use. So we refer to the I still can't figure out how you get the color on there without. All right. So he referred to when you start kind of a cross. So he's crossing things. Essentially, he has 2 plants. You start off as these are the parents. So the parents are referred to as the P generation, right? And then you take the parents and you cross one parent with another and you produce offspring. And so the first set of offspring is called the F1 generation. So F1 generation is the first set of offspring produced by crossing parents. By crossing, I mean by, we're talking actually about pollination here. So it's cross pollination. So you take the pollen from parent 1 and you use it to fertilize the flowers from the second parent, and you take the pollen from the second parent and you use them to fertilize the flowers from the first parent. You cut off all the pistils and stamens and stuff on each one of them, and then you're able to control that they don't self-pollinate at all. And then we would have an F2 generation, so that would be our second generation if we needed to cross any of the offspring from the F1 generation, and so that's kind of what they're done. And so it's able to do a couple different kinds of crosses, a cross called a monohybrid cross and a cross called a dihybrid cross. So I'll put this down below here. A monohybrid cross is where we are just looking at one trait. So okay, and we use a Punnett square to do our monohybrid cross. So I'm going to show you a picture here. Here's our picture. This is on your note sheet as well. So in this particular trait we're going to be looking at is seed color. So we have our seeds, yellow and green. And in this cross, the parents are both true breeding or homozygous. Our first parent has a yellow seed, and as we can see, yellow is dominant here. So our yellow seed is dominant. We know this because we're using a capital Y to represent the fact that yellow is dominant. There are two capital Ys, so this is telling us that this is homozygous or true breeding. Each one of these capital Ys represents one of the alleles for that particular parent. So that parent has two alleles, each one is a capital Y. Then we have our second parent, which is a green seeded parent. And again, this is a parent that has homozygous for that green color. And we represent the allele for the recessive allele as a lowercase y. Notice we don't use a G for this. Some people would want to use a G to represent green, but we're always going to use the letter for the dominant characteristic. You always use a letter for dominant characteristic. You use uppercase or the capital letter to represent the dominant one and the lowercase to represent the recessive one, okay? So when you do a Punnett square like this, you are going to use 4 squared grid, and you're going to place one of the alleles for one of your parents above the top part of the grid, and you put one here and one here. And then for our other parent, you're going to put the alleles here and here, okay? So it's then it ends up functioning a little bit like a math problem. I think it kind of reminds me a little bit of multiplication of the distribution property, right? So how we fill this in is you're actually going to, let me see if I can do this. All right. You're going to take this uppercase Y and you pull it down into this section of the grid here. And I always try to be really, really deliberate about writing my letters lowercase and uppercase. Otherwise, sometimes I confuse myself if I make them too similar to each other. And then this little y gets pulled over to the grid as well. So you take one from the top and one from the side, and where those two meet, that's what goes in that space of the grid. And then you do that also on this side. You pull this y over and this y down. Okay? I'm just going to get rid of those little arrows because they were kind of fat. Oops. I want to get rid of that one. Okay. So again, big Y, little y. You do that for the bottom as well. Again, this guy here goes all the way down, and this one goes over. So you're basically having the same in all 4 grids for this particular mono-hybrid cross. So what this tells us, again, this is our P generation, here and here. Those two are our parents, right? So our P generation, I could write this a slightly different way. Sometimes you see it written like this. Sometimes you see big Y, crossing with little y. And that tells us what our cross is going to be. And then this is our P generation. Ooh, that's a really fat letter. That's our P generation. And then we need our F1 generation. And so our F1 generation is going to be the offspring of our P generation. And here we have our 4 different potential offspring, all of which have the exact same genotype. All right, so got a couple of terms here we have to go through. We have genotype. Genotype is the combination of genes. The genotype for everybody in the F1 generation here is all big Y, little y. All of them are big Y, little y. So I'm going to put that's 100% of my offspring from my F1 generation. My phenotype is what do these guys look like, or what characteristic are they actually expressing? And because I have to follow the rules of dominance and dominance and recessiveness, right? So if you remember, that means that even if they have a recessive allele, they're not going to show it. They're only going to show the dominant allele. Because each one of these guys are big Y, little y, they're not going to express that little y trait. They're only going to express the big Y trait. So they are all yellow. So my phenotype is 100% yellow. So I can look at the percentage of genotype, the percentage of phenotype, and it's not always going to be 100%. This is just because we're doing just a homozygous cross to a homozygous cross. You end up with 100% of everybody in the F1 generation has the same characteristics. Okay, so that's what we call a monohybrid cross. There is a second type of a cross that you can do called a dihybrid cross, and it's a bit more complicated, but it's the same kind of process. So first of all, I'm going to put this up here, and again, these are all on your note sheet. I'm just looking at this because this is all of our different characteristics that these pea plants have. You can have, let's just back up a little bit here. You can have seed shape, round versus wrinkled. You could have seed color, yellow versus green. You could have flower color, purple versus white. Pod shape, inflated versus constricted. Pod color, yellow versus green. Flower position, axial versus terminal. This means the flowers are kind of like, I call them the armpits, but it's like where two branches come together. It's the axes, or kind of an armpit. Think about if you have two different branches here. If my pen's a branch, and my arm's a branch, then the flower will be right at the branch point, which is called axial. Or you can have stem height. Are they tall versus dwarf or small? So with a dihybrid cross, what we want to look at is two different characteristics at the same time. Oh, no, I'll bring that down here, but then I got to zoom back in. Okay. Kind of teeny tiny, but we'll get to it. So we're going to look at two different characteristics. So let's say we have, I don't know, I'll just make one up here. Let's say we have a parent who is dominant, heterozygous dominant for yellow pods. So yellow pods like this. That's our first parent. There's parent number one. That's our pod color. He's heterozygous. I'll just say heterozygous. There's no dominant involved. It's just heterozygous for pod color. So has one allele for yellow pods, one allele for green pods. Remember, you can't mix the letters, so we use the capital Y for the yellow pod and the lowercase y for the green pod. Our second parent is homozygous dominant, so yellow pod all the way through, big Y, big Y. Our first parent has purple flowers and is dominant, homozygous dominant for purple flowers. And our second parent has purple flowers also, but is heterozygous for purple flowers. So those are our two genotypes, parent number one, parent number two. Okay. So the first thing you have to do is figure out all the potential combinations for each parent. So there's an extra step involved here. Okay. And it's actually pretty easy, but you just got to remember to do it. People oftentimes forget this step. So when fertilization happens, you're only able to transfer over one of your alleles for each gene. So either parent one is going to donate the big Y or the little y, but he can't donate both, just one or the other. And either parent two is going to donate one of the two big Ys. So they're both the same, right? So our potential combinations for parent 1, he or she is a plant. I don't know if there's genders assigned to plants here, but it could be a big Y, big P. So we do like that. It's kind of like distribution again. So we can do that combination. We could do this combination. So that's in there twice, really. Big Y, big P. We could do this combination here. Oh, man. So that's little y, big P. I always write them in the same order every time, though. So little y, big P. Or again, you could do this combination, little y, big P. Right? So there's 4 potential combinations for each one. There's always going to be 4 potential combinations. When we look at our grid, whoopsies, there's 4 boxes going on the top and 4 boxes going down the side. So that's where those 4 different combinations are going to go. And you do the same thing with this one here. You're going to go big Y, big P, big Y, big P, big Y, little p. Oh my gosh. And big Y, little p. All right. So that's where those combinations are going to go. All right. So zooming on out here, I'm going to place the combinations on the top and the side. So all the combinations for this parent here, we're going to put on the top. And I'll use a different color. So I might zoom in because I don't know if I can draw this well. So big Y, big P, big Y, big P. All right, let's see if I can do this. And then little y, big P, a little y, big P. And we do the same thing on going down. So these guys are going to go on the left side here. Big Y, big P, big Y, big P, big Y, little p, big Y, little p. All right. So once we have all of our combinations, then we're just going to do the same thing we did with our monohybrid cross. We're just going to bring them down and over, right? So we end up with big Y, big Y, big P, big P, big Y, big Y, big P, big P, big Y, little y, big P, big P, big Y, little y, big P, big P. You just keep doing this, right? I know my writing is horrible, but so there we go. We have a big Y, little y here. Yeah, it's a little bit time consuming, but it's really not that difficult. Big Y, little y. It's the only one that has that combination so far. Big Y, big Y, big P, little p, big Y, big Y, big P, little p, big Y, little y, big P, little p, and big Y, little y, big P, little p. All right. So once you have all that information, which is a lot, you can now figure out your genotypes and phenotypes as well here, right? So what we can look at is, for example, we can look at this guy. This guy is going to be yellow pods with purple flowers, all dominant, all homozygous dominant for both, right? And that's the same thing for this one, and for this one, and for this one. And I think that's all of them. So 4 out of 16, which is 25%, are going to be homozygous dominant for both traits, right? So that's kind of how you do this. You figure out the number of them out of the total, which is 16, that have that combination of genes, and you can figure out the ratio of that genotype. Remember, phenotype is the coloration. So we might say, well, how many of them have a yellow pod? Well, 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12. They all do. All 16 do. Because even those that are big Y, little y are still have that dominant allele, so they have to be yellow podded. So we'd say 100% of them have a yellow pod. Same is true, 100% of them have purple flowers. So the phenotypic ratio, the phenotype ratio is going to be different from the genotype ratio. All right. Well, that pretty much goes through everything, you guys, in this lesson. If you have any questions, feel free to give me a call or an email. Otherwise, that's the end of this one, and I will see you in the next.